And our next speaker is going to take a more holistic view here, John Storkey. He's going to tell us about a systems-based approach to crop protection. John, you've got this chair.
Thank you. Good morning, everybody. So yes, as the last talk proper in this session, I'm going to pan out a bit and take a more holistic view of farming systems in the context of crop protection, but hopefully pull together some of the threads that we've been thinking about from the various talks this morning. And my starting point is that we've become overly reliant on chemical crop protection products, and that has created problems that are now working against us. Negative impacts on non-target organisms, for example, and the evolution of pesticide resistance. And that's has been a consequence of the fact that the ease of use and efficacy of those products has meant that our farming systems have become oversimplified, I would argue, which means they've become less resilient to those pest, weed and disease problems starting in the first place. So what we need to think about, as well as how we react to those problems when they occur, is how we build systems that are more resilient to them. And so this slide just illustrates that simplification of our systems at three scales. On the left, we're now growing a smaller range of crop cultivars for any given crop. We tend to look at the recommended list and pick the top yielding ones. Granted, we think about resistance to diseases, but generally those cultivars are bred for yield in conditions where we use crop protection products, so they're ideal conditions where we can control the crop pests. And so we've lost some resilience at the population level. At the field scale, fields have become bigger. We've lost semi-natural habitats around fields and habitats for natural enemies, as Sam was talking about. It's true that we've started to reintroduce hedgerows and field margins, which has helped with that over recent years. But also on a temporal scale, our fields have become more simple in terms of the number of crops that we grow, and our rotations are now shorter, which means we use a smaller range of active ingredients, which increases selection pressure for resistance. And finally, on the right there at the landscape scale, as our systems have become more specialised, so we tend to specialise in arable or livestock, and so we have this separation of the different systems. The landscapes become simpler with less fragmentation, and that tends to select for generalist species that are able to disperse easily, and it could be argued are more adaptable to control strategies. And again, there's a loss of habitat for natural enemies. Okay, next slide, Laura, thank you. So the response to that is to think about how we re-diversify our systems. And what we're not talking about here is going back to pre-war agriculture and throwing the green revolution baby out with the bathwater. As we've tried to emphasise as we've gone through this morning, we think chemical crop protection products will remain a really important tool for the future, but we want to develop ways of using them in a sustainable way. And that will involve, at some level, re-diversifying the system. So what I've tried to do here is compare an approach or a strategy that's very reliant on pesticides with one that takes more of an integrated pest management approach, which hopefully reminds you of some of the things that have been said already. So a chemical crop protection strategy is very reactive. We build a system for profitability or for yield, and then when a problem pops up, we look in the green book and we find the active that controls it. IPM thinks about how we build that system that prevents the problem emerging in the first place. Crop protection strategies are often restricted to a field scale and a single season, whereas IPM thinks across the landscape and across the cropping system. The traditional approach to crop protection focuses on the individual target species, whereas IPM seeks to manage an ecosystem and those trophic interactions. And then finally, as we've talked about before, we're seeking to complement chemical control with non-chemical control. Thanks, Laura. So all of that as a background creates a challenge to our conventional approach to research that we take at Rothamsted in terms of how we look at this issue of a systems-based approach to crop protection. We can no longer just take a single input, like, for example, glyphosate, and look at the effect on a single target, for example, blackgrass, and adjust the doses or adjust the timing. We're more interested in how different parts of the system combine to explain the variance in pest, weed and disease pressure. So perhaps we need a different approach to agricultural experimentation. And for the rest of my time, I'm going to introduce a new experiment at Rothamsted, which our ambition is that it becomes a new long-term experiment, and we call it the large-scale rotation experiment, or the LSRE. And crop protection is an important component of why we're doing this. Thanks, Laura. So to introduce the experiment, we've started it at 2 sites, at the research farm at Harpenden and at Brooms Barn. It's a large experiment. It covers 9 hectares at each site with 60 24 by 24 metre plots. Those plots are split into 2. Half of them are ploughed annually, and the other half we're aiming for a zero-till system, but at the moment we're just reducing the tillage and we're in that transition phase towards a zero-till system. We then, on that cultivation treatment, we have 3 rotations with increasing crop functional diversity. So at the top there we have 2 wheats and oilseed rape. Then we introduce in a 5-year rotation field beans and spring barley, and then in a 7-year rotation we have a 2-year short-term grass clover lay, and we replace the oilseed rape either with sugar beet at Brooms Barn or linseed at Harpenden. And then each of those main plots is split into 2 with organic amendments on one side and just inorganic fertilizers on the other side. Next click please, Laura. And then over all of that we have a smart crop protection treatment. So half of the plots in all of those combinations of treatments are managed according to IPM principles, the others conventionally. Thanks, Laura. So this is an illustration of the rotations that we're using. We've tried to aim for things which are agronomically realistic, but I need to emphasize at this point that what we're not trying to do is design the ideal system. What we're trying to do is create management contrasts that will allow us to see how individual parts of the system, for example, weeds or pests or diseases, respond in the context of those different systems. So you'll see here the 3 rotations. We broadly call them economic or short-term economic gain for the 3-year, agronomic sustainability the 5-year, and aiming for environmental goods and services like soil carbon in the 7-year one. Thanks, Laura. So a slide on the smart crop protection treatment. So again, we thought about individual interventions you can do in a given crop, oilseed rape or winter wheat or whatever it is. And we are doing that in a crop specific way. But remember that these, I could call them secondary interventions, are being done in the context of those different system templates. So we'll be able to see the relative effectiveness of diversifying your rotation compared with, for example, companion cropping. So these are some of the approaches that we're taking on that smart crop protection treatment. We're looking at choice of cultivars, cultivar mixes, delayed drilling for blackgrass control, companion cropping for flea beetle control, and spraying to thresholds. And as well as looking at the impact on the pests, weeds and diseases, we're looking at pesticide use and the economics of those approaches as well. Thanks, Laura. So I don't expect you to digest this slide at all. It's just here for illustrative purposes to show you that we are measuring lots of things on the experiment. So as opposed to looking at the effect of one treatment on one thing, blackgrass or blackgrass resistance or cabbage stem flea beetle density, we're measuring all of those things, or as many of those things as we can, including metrics to do with soil health, soil microbiology, earthworms, crop physiology, crop yield, and the pest, weed and disease pressure, to understand how those things interact and how the different systems regulate the different parts of the system. So, for example, there was a question to John earlier about the importance of crop nutrition for disease management. This experiment will help address some of those questions that are to do with linking different parts of the system. Thanks, Laura. So my last 3 slides, I think, just illustrate this from a weeds angle. which tends to be my primary interest at Rothamsted. So in 2017 and 18, we did some baseline measurements. The experiment started at Brooms Barn, which is the picture on the left here, in 2017, and Harpenden in 2018. And so we have data on baseline data on the soil metrics on pests, weeds and diseases, the background problem, if you like, that we are selecting against. And so on the right there, you can see the relative proportion of different weed species across the field. In the baseline year, it was dominated by crop volunteers, the volunteer oats in purple and the orchid rape in yellow. Thanks, Laura. An important principle here is that often your trajectory towards sustainable crop protection or your IPM strategy will depend on your starting point and the particular pest, weed and disease communities that you're seeking to manage and regulate. And this slide just illustrates that at the two sites we have very different weed communities, and so our approach might be different to managing those communities. At Brooms Barn, the weed community is dominated by broadleaf species with a high proportion of spring or generalist emergers because of the high proportion of spring crops in the rotation and the light soils. At Harpenden, it's dominated by blackgrass because of the winter wheat dominated rotation and the heavy soils. Thanks, Laura. And so that then cascades through to how those weed communities are responding to the different systems. And so I don't have time to talk in detail to this slide, but if you can just quickly see that it's a comparison between the zero till and the ploughed system, and the two sites are responding in contrasting ways because of the different starting weed community. And so we'll be able to see the interaction of the different species pools, the systems and the integrated approaches to the pressure of pest, weeds and diseases and pesticide use. And that's my last slide. Thank you, Laura. I think there's one more thank you slide. Thanks very much.
Okay, thank you very much, John. The LSRE is a really exciting new venture for Rothamsted, and I think it will hopefully get established in the way our well-known long-term experiments are very useful to a whole range of science and growing conditions. We've got a couple of questions already, I hear. So this is an anonymous one. With the LSRE project, with so many variables, how are you going to find significant effects attributed to a singular change in land or crop management?
It's a really good question, and actually it points to the fact that the statistical analysis we're going to use is slightly different. So yes, we do have some power in the experiment to say if you change the cultivation or if you change a particular pesticide regime, how will a response variable change? But that's not the primary way that we plan to analyse the experiment. Rather, we're looking for gradients. So there'll be a gradient of, for example, soil carbon or a gradient of soil porosity. And then there'll be a gradient of blackgrass density or blackgrass emergence. And it's relating those gradients to each other. And that we think will be the primary way that we will analyse the experiment. So we're seeking to understand how the systems behave and how important, for example, soil health is in determining weed pressure or disease pressure.
Interesting. Another interesting question too, which I hadn't thought about. Is this large-scale experiment linking into any other UK or Europe projects that have similar goals and assessments?
Ideally, yes, but I've just had a European proposal rejected, which was asking,
it's getting harder, isn't it, to get European money?
Which was doing exactly that. So I had colleagues in France with similar experiments, Denmark, Finland. So we pulled together, I think, half a dozen of similar experiments, along with farmer networks of the type that David Comment was talking about, so you could upscale the results. Hopefully those ideas will come to fruition. But excellent, yes, that's what we need to do.
Okay, and there's a final question I'm going to take here because it's Andy Barr, and I think a lot of people will be agreeing with him. Loving the LSRE, where can we find more information and results, please?
Yeah, thank you. So if you know Aisling Pearson, she's been very involved in the LSRE over the past year or so. She tends to use Twitter a lot. So again, if you contact me or someone at Rothamsted, will be able to point you in her direction. But we're just now at a stage where we can start analysing the experiment. So we're coming to a point where both sites, we've got to 3 years worth of data. We're still in the transition phase, but where are we now? Yeah, by the end of this year, you'll start seeing some results being promoted.
Okay, so hopefully that'll be scientific publications and more general information for growers and farmers, John. Yeah, that's great. Thank you.